Hello everyone and welcome to SenzaGen's poster presentation. My name is Andy Fogeryd and today I will be talking about the potential to perform quantitative skin sensitizing potency assessment of chemicals in the GARD skin assay. So the field of toxicity testing has recently undergone a paradigm shift to move away from animal experimentation and instead promote the use of in vitro assays. So today several non-animal based assays for hazard identification of skin sensitizer have become available, but despite this progress, there is still no in vitro alternatives that are capable of providing a reliable assessment of the sensitizing potency. And this is really important for quantitative risk assessment and in order to derive safe levels of exposures. So the GARD skin assay brings novel elements to the field of regulatory toxicology by evaluating the gene expression changes across a biomarker signature of 200 genes involved in pathways associated with skin sensitization. So following the exposure of the cells to a test substance and the measurement of gene-based biomarker signature, a machine learning algorithm is applied in order to assign a decision value to each of the test items. So a positive decision value by the algorithm is classified as a sensitizer and a negative value is classified as a non-sensitizer. And this assay has proven to be very accurate and is now currently under review by the ECVAM for inclusion into OECD test guidelines. So the aim of my poster today is to present a novel testing strategy for assessment of quantitative skin sensitizing potency on a continuous scale based on the output from the GARD skin assay. So this figure briefly summarized the experimental protocol that were applied in the poster. So the GARD skin assay were performed in a titrated range of concentration instead of only at one concentration. So the top concentration used was 500 micromolar. The standard GARD skin protocol was implied in order to generate decision values for each of the concentration and for a total of 29 substances. And those response curves were then constructed for each of the substance by plotting the decision values versus the input concentration, and linear interpolation was finally used in order to estimate the lowest concentration required to induce a positive classification as a skin sensitizer in the GARD skin assay. And that is here represented by a positive decision value. And this concentration is what we refer to as the CDV0 concentration. So in this figure, the dose response curves for all substances have been summarized and colored according to potency as defined by the CLP categories, where strong sensitizers in pink, weak sensitizers in blue, and non-sensitizers in green. So only two of the non-sensitizers gave rise to a positive CDV0 value, but these were compounds that were assayed at a concentration above the maximum input concentration in the assay, so they were indeed correctly predicted as non-sensitizers. So for the skin sensitizers, all except two gave rise to a nice dose response curves, and the exceptions were in this case were very weak sensitizer, which by definition would require a much higher concentration in order to induce a positive response. And in this case, they were unfortunately limited by solubility issues, but we will further explore the testing of these substances using less polar solvents in order to be able to increase the concentration. So in order to conclude the information from the figure, we can see that strong sensitizers triggered response signals above the binary threshold at much lower concentrations compared to the weak sensitizer. And we can also see that there was almost a perfect separation between the CLP categories. So moreover, by looking into how well this approach can provide continuous quantitative potency assessment, the CDV0 concentrations for all sensitizers were compared both to the local lymph node assay EC3 values in the figure to the left and to the human potency categories as defined by David Basketer to the right. So we can see that there was a significant correlation between the GARD CDV0 concentrations, both to the local lymph node assay EC3 values as well as to the human potency categories. So this would enable for a direct extrapolation between the different measurements as illustrated in these examples. So once you have generated a CDV0 concentration in the GARD skin assay, this value can then be used in order to predict a corresponding local lymph node assay EC3 value or a human potency category. So in order to summarize the main conclusion from this poster, we here introduce the GARD skin dose response assay as a suitable tool for quantitative skin sensitizing potency assessment of chemicals. And we also propose a protocol that can be used as a tool for risk assessment of chemicals and mixtures without the use of animal experimentation. And the experimental readout that we use, which is here referred to as the CDV0 concentration, correspond to the lowest concentration required to elicit a positive response in the GARD skin assay. And these experimental protocols are in analogous to the local lymph node assay, in which the CDV0 concentration correspond to the EC3 values. The GARD skin dose response CDV0s may be used directly to monitor the sensitizing potency of a compound, or it can further be used in order to extrapolate local lymph node assay EC3 values, or for estimation of human potency categories, or also for direct assignment into different CLP categories as strong and weak sensitizers. So thank you all very much for listening. I will be really happy to take any questions. And also please feel free to reach out to myself or any of my colleagues at SenzaGen at any times if you have any question relating to the material in this poster. Thank you very much.